This is our confocal microscope, or to give this proper name, our laser scanning confocal microscope. You'll see why it's called that in a minute. Ours is made by Leica, and it's built onto an inverted microscope frame. So think of a normal upright microscope, turn the whole thing upside down. There are good reasons for doing this, as it lets us look at a much wider range of sample types than a conventional upright microscope would allow us to. It's surrounded by a heated and insulated box, which allows us to keep living samples warm and happy during imaging.
Here is how the microscope produces an optical section by eliminating all out-of-focus information. As we've already seen, in a conventional microscope we capture information from all levels in our sample, whether they are in focus or not. So here we're focused at the midpoint of the cell, shown in red, but we still see out-of-focus signal from above and below. To eliminate the out-of-focus signal, we place a pinhole between the sample and the detector. Only light from the focus level gets refocused at the pinhole and can pass through it to be detected on the far side. All out-of-focus signal hits the plate around the pinhole and is rejected.
The microscope uses lasers of different wavelengths, in other words different colours, to excite the fluorescence of the probes which we stain our samples with. Unlike a conventional microscope, where the whole field of view is filled with light all the time, in the confocal microscope we focus the laser light into the smallest possible spot. Then we use a pair of oscillating mirrors to deflect that spot left and right and up and down. By coordinating the movement of these two mirrors, we can drive the spot in a scanning pattern across our sample. Then, if the laser hits a fluorescent molecule that it can excite, the fluorescent molecule will glow, give out light. The fluorescence, which is a longer wavelength, so a different colour, passes back through the microscope's optics and is separated from any reflected laser light. The fluorescence then hits the pinhole and any out-of-focus information gets blocked. The in-focus information passes through the pinhole and is detected on the far side using a special detector. Because we're using a spot of moving laser light, we know exactly where that spot is on our sample at any one point in time, and therefore we can map the brightness of the fluorescent signal it generates to an individual pixel in the image which we create.
We have 4 detectors on our system, which are individually tunable to detect any colour ranges that we want. So we can image up to 4 different fluorescent probes targeting different things and that glow in different colours all at the same time. That's both really cool and really useful. Let's look at the system in action. The sample we're going to look at is a small, shrimp-like animal from Southampton Water that was caught by the Oceanography department. As you can see, it is only a few millimetres long and it is very three-dimensional. It has been stained with red food dye to make it fluoresce, and then made transparent by a process called optical clearing. We need to do this to get the laser light into the sample and to let the fluorescence escape to be detected. If we look at the sample by normal fluorescence microscopy, and this is just part of the head at about 200 magnification with the eye, we can see fluorescence coming from all levels in the sample at once, whether they're in focus or not. So it's really hard to see and understand the image and what's going on. So, how do normal and confocal imaging compare as we slowly focus through our sample? We are going to use 3 different confocal detectors, one here, one here, and one here, to look at 3 different colour ranges of autofluorescence in our sample at the same time. In the blue range, in the green range, and in the orangey red range. So this image here is not a confocal image, so we see the in focus and the out of focus at the same time. If we then bring in the signal from our confocal detectors, the first one, the second one, and the third one, we can see that our confocal fluorescence is only coming from one level in our sample. Now obviously we can then set the microscope to take a whole series of images at different focus levels automatically.
Once we have acquired our series of images, there's lots we can then do to manipulate the data in different ways.
We can also squash all of the individual slices down into a single image, showing the in-focus signal from all levels in the specimen in one go. Having collected the images from all the different focus levels, we have a 3D data set, and that then allows us to look at the data from any angle we want, simply by letting the computer do the maths on the dataset to calculate what it would look like from that angle. We can also view images from the side, so as well as the XY plane in which the images were captured, we can look at the XZ and YZ views. I think you'll agree, it's a great bit of kit.